Right. Okay, beta.
Yes.
So today our topic is the mitotic cycle.
Yes, sir.
Mitotic cycle. Uh, Mitotic cycle. It all first we should know its importance.
Yes, sir.
Why our body or any organism needs this cycle, this mitotic cycle.
Yes, sir.
Hmm. As we know, every cell has to divide because this process is very important for the survival of an organism
Yes, sir.
or the survival of any specific cell. So this mitotic cycle is, you can say, a cardinal or very important feature of any organism because it actually helps in cell division. And cell division is the basic character of any living organism because this cell division is actually the character of reproduction. Something is going to reproduce, right? So in this chapter you will understand that how this cell division is taking place or what type of steps or events which are taking place before this cell division.
Yes, sir.
So we commonly divide this cell division into two main steps. One is karyokinesis
Okay, sir.
and the other one is cytokinesis.
Okay, sir.
Are you getting? So so these are two main steps of cell division. So in this case, in mitotic cell, our focus would be on this karyokinesis or this nucleus division. We will try to understand that what's going on in the nucleus and how this karyokinesis is taking place in an organism, right? But before that, the one thing you should know that is the structure of
Yes, sir.
chromosome. Structure of chromosome. The common shape or the structure that we always observe, that is a chromosome, a body that contains two arms and one central point that is called centromere, right? So this is the common or the, you can say, a main structure of an organism of
Yes.
a chromosome, but this shape is commonly changed because chromosomes are different types, so they have different length of these arms, but typically we commonly use this shape of chromosome, right? So at the end of this, here there is a portion of the chromosome that is called that we commonly use telomere, and it is present on both sides.
Yes, sir.
Telo. Are you getting this point? Is it?
Yes.
So it is a condensed structure like this. You would have the nucleus here, but that is in a very condensed form. So we commonly use this a term that is chromated. Are you getting? Chromated.
Yes, sir.
So this is the common or the typical structure of the chromosome. But in case of mitosis or any mitotic cycle, we will observe there will be a division of this chromosome and it will be converted into a pair of chromosome like this. You would find after division like this. So these two structures, when the single chromosome further replicate to produce identical arm like this is what we call sister chromatid. This is what we call sister chromatid, right?
Yes, sir.
So another thing that you should know, as I have told you that what is this chromatid, the basic concepts that should be clear in your mind, that is chromatid,
Yes,
right?
sir.
And what this chromatid is. Hmm. Give it up. It's a condensed form of DNA.
Yes, sir.
Is it? Each chromatid is made up of DNA molecule, and each DNA molecule replicates before cell division to form identical copies of, as I have told you, that these identical copies are formed. So this chromatid is actually the condensed form of that DNA. As we know, this DNA is wrapped in the histone proteins. There are histone proteins there,
Yes, sir.
and these proteins are actually involved it to condense this DNA like this, and there will be a linker DNA which will connect these two histone proteins with each other. So this is how this chromatid is, right?
Yes.
So telomere. Now I have used another term there, that is telomere. We should know about this, what it is, why it is here. So this concept should be clear that these are the actual tips of the chromosome. Now you should be clear in this case. If you are showing this, it is a single copy of chromosome.
Yes, sir.
Whenever you see a structure like this, it is the pair of chromosome. It is the this is the copy of this one, right? So you should keep in mind. So this is a pair structure. It is not a single chromosome. It is a, you can say, a pair of a chromosome where you have the identical copy of this one. So
Pair
we
of
call it a sense. Yes, pair of chromosomes, because this is a single chromosome, and when it replicate, it produce identical arm, identical copy, that is like this. So
Yes, sir.
these are called sister chromatids,
Yes, sir.
right? These two arms, you can call it sister chromatids. So
Yes, sir.
telomere, I was talking about the telomere. Telomere, right?
Yes.
Telomere. So it is the repetitive nucleotide sequence at the end of the chromatids. What it is going to do here, what is its function here? These are present like here. This is telomere. Here you would have a telomere. Here you will have a telomere. Why it is present at the end of this chromosome? The answer is it has two functions. You can say mainly it has one function. That is, it protect the deterioration of the chromosome, or the damage of the chromosome, or the breakdown of the chromosome. breakdown of chromosome. How it is protected? Give it up. It is protected by protection. We can
Yes,
say protection. And this protection is provided by this telomere. And what these telomeres are? These are the repetitive
units.
sequence of nucleotides.
Yes, sir.
Are you getting?
Yes, sir.
All right.
Yes, sir.
Do you want to ask anything?
सर जी टीलोमेर्स करते क्या हैं?
बेटा यही बता रहा हूँ. I have told you that this
प्रोटेक्शन मतलब किस तरह?
telomere protection, because this is a repetitive structure. The number of nucleotides are present here. So just in a way that how it can protect it. So this breakdown can be possible by any external factor, like because it is the repetitive sequence. So if few nucleotides which are going to loss from here, so there would not be affect the overall structure of this chromosome. So because of the condensation of these nucleotides, same nucleotides condensation here at the tips of these chromosomes, they actually, you can say, they are involved in anti-aging. They help not to degrade or to become aged to these chromosomes. The very simple, because of this sequence, because of this repetition. Very simple, like if you have a single block here, right? And if you have a repetition of these blocks like this, like this, like this, like this. So what do you think? Is it possible to break this single block or to break this complete block? Or the number of blocks which are piled up on each other. So definitely this single block is easy to break down. Is it?
Yes, sir.
So, but you have the pile up of these, a number of blocks which are very much attached with each other, close to each other. So it is difficult to break it, right?
Yes, sir.
So this is how it protects the breakdown of these chromosomes, or these are the anti-aging. Another thing which is very much associated with it, that is, it helps to protect the loss of genetic information. Genetic information.
Yes,
Loss of genetic information, remember. So that work is same. Actually, it's providing the protection. So this protection may be like the protection to stop this breakdown, or not to let this chromosome to be broken down. And the second function is it helps this to protect the genetic material. Right? Are you getting it up?
Yes,
Achha. Achha. So chromatins, I have told you that it has its composition is, it is made of two components. This chromatid, and what are those components? G.
Telomeres and chromatins.
This is chromatid. Chromatid. This condensed structure. Chromatid. This chromatid is composed of two things: DNA plus histone protein. DNA plus histone proteins. Are you getting?
Yes, sir.
So this is the genetic information that is stored in DNA in the form of genes. You know very well genes. These are the basic unit of this complete DNA, right? So what is the function of this gene? What it does here?
Contains genetic information.
Yes, and how that genetic information will be translated?
Sir, through gene expression.
And how that gene expression will be possible?
सर ये हो
How
सकता
that will be expressed? What do you think? This gene is what is its
ट्रांसक्रिप्शन
standard?
ट्रांसलेशन से।
Okay, through transcription. It means you're saying that it would be messenger RNA.
यस सर।
Then you would have hmm, hmm. Then you had a polypeptide.
यस सर।
That polypeptide will be formed because of this translation. So
ये
transcription, translation. There is a central dogma here, right? All right. So DNA is a long molecule. It is about 2 nanometer wide, and total length of DNA is, you just imagine, 46 number of chromosomes in human cell. That is about 1.8 meter.
यस सर।
1.8 meter.
यस सर।
This 1.8 meter is in width. How many? 46 chromosomes. So this chromatin, why it is condensed here? Because in order to occupy a very small space, it is about 1.8 meter. And if you just imagine that if it is not condensed, so what would be the consequences? The consequences are it is difficult to accommodate in such a long DNA in a very small cell. Are you getting it? Is it fine?
यस सर।
And it is packed in 6 micrometer size in nucleus. You just imagine 6 micrometer, where it's 1.6 meter and it is in 6 micrometer. So this long thread is accommodated in a very small space, that is 6 micrometer. Why? How? That is
स्टोन
because of this condensation.
यस सर।
Because of this condensation, this chromatid formation, right? Are you getting this point?
यस सर।
So this was actually I wanted to tell you, and I think I have discussed all about the linker DNA as well.
यस सर।
How many histone proteins are involved here?
सर जी 2।
No, 8 histones.
ओके सर।
Eight histones. Right.
ओके सर।
8 histone proteins are there.
8।
Eight.
एक मिनट सर जी 8 का पेयर या टोटल 8?
8 histone molecules to form nucleosome. There will be a nucleosome here actually, and not pair. I'm talking about 8, 4 pairs,
ओके सर।
right? In
यस।
this case, so linker DNA. I have told you like there is a histone here, there is a histone here, and you have the wrapper DNA in this case, and we have wrapper DNA here. So this is the linker DNA.
यस सर।
Linker DNA. It is going to link these two histone proteins, and this is how this is what we call nucleosome.
यस सर।
It's a body which is formed with this DNA and histone protein. That is formed a nucleosome, right? So बेटा इधर कंक्लूड करते हैं बिकॉज मैंने तो बाहर जाना है, और कल से हम क्लासेस आपका सैटरडे भी ऑन है, ठीक है? कल फ्राइडे है ना?
ये सर जी कल
तो अब हम
थर्सडे।
जो है क्लास
कल थर्सडे है सर जी,
ओ
मेरी
ओ
बात सुने।
जी जी बेटा।
सर जी आपकी क्लास की टाइमिंग ज़रा थोड़ी सी अजीब है, क्योंकि कल से मेरी फिजिक्स की क्लास भी 7:15 तक होती है, और
हाँ
10
हाँ,
तक तो मेरी वो दूसरी केमिस्ट्री क्लास होती है, तो ये आप एट ओ
तो हम
क्लॉक
अपने टाइम से कर लेंगे ना। टाइम आप इसको बेशक अब इट डिपेंड्स ऑन यू, आपकी ईज पे है। आप जिस टाइम लेना चाहते हैं लेट मी नो अबाउट दैट, मैं उस टाइम पे उसको एडजस्ट कर दूँगा, ठीक है?
सर जी 8 पे नहीं हो सकती?
हम लाभ कैसे ले रहे थे? एटीपी ले रहे थे पहले।
हम जी 8 ही ले रहे थे।
इट्स ओके, इट्स ओके। तो फिजिक्स कितने दिन हो रही है बेटा?
सर जी फिजिक्स तो होती है ना वीक में, मतलब 3-4 दिन होती है।
हाँ, 3 days। चलो बेटा, फाइन फाइन, इट्स ओके।
ओके
ऑलराइट, हम 8 o'clock कर देते हैं, और फिर जब भी हो आप सैटरडे संडे को फ्री, तो मैं आपको बता दूँ।
ओके सर।
कोऑर्डिनेट कर दिया करें कोई भी टाइम जब आपको लगे कि इट इज फिजिबल फॉर मी, सो कोऑर्डिनेट कर दिया करें सो दैट आई कैन देख। ठीक है बेटा?
ओके
तो
सर।
इसको खत्म करें, और फिर आप मुझे वो जो आपके पास सारी वीडियोस पड़ी हैं वो मुझे आपने सेंड कर दी है, ठीक
ओके
है?
ओके सर।
काफ़ी आप मुझे नहीं दी, राइट?
मैं सर जी मैं भेज दूँ।
ये मैं भेज देता हूँ, मस्ट
सर जी वो आपने रिमाइंड नहीं कराया, उसके बाद आपने बोले नहीं था, वो मुझे याद ही नहीं रहा।
हाँ बेटा, बस वो आप कर दीजिएगा ना।
मैं
राइट।
सर जी। ओके।